absolute pleasure to be presenting here at the first symposium, and it's a major milestone that everyone has made. So for today's agenda, I'll be giving a brief overview of Perspectum, just what we do, and our products, just to link it all up together. Conventional MRI and superficial siderosis, quantitative MRI and its application, and what's currently happening at Perspectum with regards to superficial siderosis. So Perspectum is a precision health technology company where we deliver advanced imaging solutions, as you can see here on the right-hand side. We deliver both pharma solutions and clinical solutions. So with regards to pharma, we try and deliver faster clinical trials. And for clinical solutions, we deliver advanced diagnostic tools for better care for patients so that doctors can really know what to do next for their patients and hopefully ensure better outcomes. So here is just a few things that we do. We do fat quantification as illustrated here. So you've got the heart, kidneys, and lungs as well. And then we also do fibroinflammation quantification. Again, here are some of the organs that we cover. And then in addition to that, we do iron quantification on the liver. So to do this, we have 4 products at the moment. MRCP Plus, which kind of looks like this on the image, which is magnetic resonance cholangiopancreatography scans. And this is where we visualize and quantify assessments of the biliary and pancreatic duct. Next, we have Cover Scan, and this one assesses a range of multi-organ tissue characteristics, the size and their function. And this one in particular covers the heart, lungs, spleen, pancreas, and kidneys. Now we've got Hepatica coming up next. And this one is, I quite like this one because it helps make decisions, pre-surgical decisions for patients that have liver cancer. And then finally, we have our flagship product, which is Liver Multiscan. I'm not sure if any of the clinicians have heard about this one, but it has been drummed up lately in the pharma space. And this one in particular has 3 metrics. So we've got a proprietary imaging biomarker, corrected T1, better known as CT1, and this one measures liver disease activity predictive of liver and cardiovascular outcomes. We then have liver fat fraction measured with proton density fat fraction, which is considered the gold standard in measuring liver steatosis, which is your fatty liver disease essentially. And then finally, we have liver iron concentration, and this is a measure of iron present in the liver reported in milligrams of iron per gram of dry tissue. Now, with this in mind, an image can make the impossible possible. And that's my personal opinion, and make the invisible visible. So as you have on the liver, why can't we do it on the brain? And this is where we can hopefully apply this to superficial siderosis. And we are often left seeing the damage without really having a way of measuring it and predicting where it's all heading. And in superficial siderosis, that accumulation is silent, and the iron ends up devastating the brain and spinal cord. And until recently, like with Liver Multiscan, there isn't really a way of truly quantifying it. So before I go any further, I want to share for those that don't already know, I'm speaking to you not just as a researcher and project manager, but also as a patient living with superficial siderosis. So this, in fact, is my own brain scan. And for me, it's not just academic, it's a journey to find a potential solution. And it's one of the reasons why I believe developing tools like the ones that Perspectum has, that we can apply that to superficial siderosis and then track iron burden. And it's not just scientifically interesting for me personally, but I think it's crucial for patient care. This photo was taken back in around 2003, which is just before I had surgery to remove a subarachnoid cyst, which is basically the cause of the low volume bleed that has led to superficial siderosis. And of course, now it's 2025, so it's now over 20 years since that operation, and it's led to many questions, such as, does everything need to be so invasive? Do we need to have repeated lumbar punctures or myelograms? Why can't we just not be prodded around with needles? So this does really apply, not just applied to superficial siderosis, but to other neurodegenerative diseases. And from the talks that occurred yesterday, there is this massive unmet patient need. And for myself, it's that gap between 2003 and 2016 to when I was diagnosed with superficial siderosis. And for all those years, there wasn't really anything going on, maybe a little bit of tinnitus, but nothing more to that until 2016, when I had quite impactful symptoms. And then that's when I received that diagnosis. And then you, as patients, you have that need to go look for more answers, go Google it, figure out what's going on. And back then, I don't recall there being much out there. We had those couple of Facebook groups, but nothing more to it. And then because of my degree, I was actually in first year university when this all happened, I decided to just keep going and looking into the research and seeing what we can find to better the community. And yes, that did lead to questions of, do you take the ferroprone? Do you take the surgical option? What do you do when you're reading the statistic at the time that you're 1 in a million? And that, for everyone, I'm presuming, was quite daunting. So today we'll explore how advanced MRI techniques are finally giving us the way to quantify and hopefully change the trajectory of neurodegenerative conditions. So here we have T1 and T2 weighted MRIs, just to start off with the basic stuff, basically. And the difference between these two is that T1 weighted enhances the fat, which is the brightness that you see, and T2 enhances the water. So basically, you want to think of it that T2 is good for looking at the overall structure of the body, and T2 is good at looking at the inflammation and fluid. And the type of intensity that you see on the scan really depends on the MRI sequences that you use. And we have the options of T1, T2, T2 star, for instance, and susceptibility weighted imaging. For superficial siderosis, the preferred option is T2 star and susceptibility weighted imaging. And we, as a patient group, are really reliant on these conventional MRI scans. But the majority of scans probably show no change, right? Because you're relying on those observational results. You're relying on your clinicians to be able to detect the tiny changes. And what has stuck to me since meeting with a neurosurgeon is that hemosiderin is like layers of paint. You can't see each individual layer, but each individual layer of it could be doing a lot of damage, but you just don't know. So here we have a conventional MRI of superficial siderosis. I believe this image was used yesterday as well. And it's indicated by those white arrows that show a dark hypointensity, which is your hemosiderin deposition. And that is what is used as one of the criteria to diagnose superficial siderosis. And that rim of low signal covering the surfaces of the brain, brainstem and spinal cord is what you would usually see. And this will vary in thickness depending on who the patient is. And then that kind of illustrates what the level of hemosiderin deposition is. Now, the crucial thing with this, again, is that it's all observational. So how is brain iron currently measured? Well, at the moment, we have iron-sensitive MRI sequences, so the T2 star and SWI are those preferred ones. You then have the appearance of specific patterns and hallmark features. So I've taken an example of this image here in a patient with pantothenate kinase-associated neurodegeneration. You see this eye of the tiger sign, that in the middle, that basically that blurring out of that shape, and that's what clinicians would, in this case, look for. Then you have the comparison of images over time. So this is also a good example here with PCAN, that you're looking at age 4 to age 10 and looking at those differences between them. Finally, you have the comparison of regions. So in some research papers, they do a comparison of, say, the red nucleus compared to the globus pallidus, and looking whether there's any differences there. And then there are additional indicators. So you'll have cerebellar atrophy. So with us, that would be the reduction of your cerebellum or the cerebrum as well. And then you have the thinning of other regions. But again, this is all observational. So the challenges with this is that there are a lack of consistent methods. Every clinician will look at a scan differently, so that there is a dependency on a clinician's experience. If someone hasn't, in our instance, seen superficial siderosis before, what are they looking for, right? And then there's quite a number of papers that have quoted this, but individual interpreter bias. I'm guessing the doctors may bicker as to what they see on a scan, and yeah, there ends up being this inconsistency of methods. Then what I've seen in my own work with Perspectum is that you have a varied acquisition of data. You have, at the moment, I work with 3 different scanner types. So Siemens, GE, Philips, and then within those scanners, you have different models, and all of those would need some level of standardization. If you take an image, say, from a GE scanner, it may look slightly different to a Siemens scanner. And then finally, there's that lack of quantitative data, and that is something for us, and this is a strong opinion of mine, that we definitely need to overcome. To do this, it would use something called quantitative MRI. Here I've just shown an image of what quantitative susceptibility mapping looks like. This is one of the methods that you can use. And initially what happens, this is quite a simplified diagram of it. It is rather complex, but what you would initially do is acquire an MRI scan. For this particular case, it's been acquired using gradient echo sequence. You obtain this and capture both two things, the magnitude and the phase. And the phase image in particular has a subtle information about tissue composition that you can extract from there. And this phase image has these distortions to local magnetic field, which is the key thing in quantitative susceptibility mapping. You're looking at your magnetic susceptibility of a tissue. So you then move on to something called phase unwrapping, and this is where this phase data is something that is obtained between the regions of pi and minus pi. And what happens when you acquire this image, so you have these jumps, you have those slices. Because they're in a specific range, they will jump from one to another, and you want to smooth it out so that you don't have those gaps. And that's where you basically smooth it out. And so if you take the idea of a telephone cord that's all tangled up, you just want to straighten it out. So then you go on to something called the tissue field, where you remove the background data. And that you can see that in the first image, all that blurring around that spherical shape, which is your brain essentially. You want to remove that data so that you are left with what you have on the brain. So once that's removed, the next step is, as I said, removing that magnetic noise, and then you combine both the magnetic and phase data back together like a puzzle through something called dipole inversion. And this is where you're looking at what is giving off that magnetic susceptibility, and you want to pick up the ion. For us, it would be the ion, and you do that through dipole inversion, and then that will give you the data for each voxel within an image. And a voxel is essentially like a 3D cube, and your image is built of these little 3D cubes. And in this process, it will give you a unit of parts per million, and that will end up being your magnetic susceptibility. Now, how has this been already applied clinically? Well, this is, again, a research paper, but this has been applied to beta thalassemia major patients. And the reason why I chose out this particular paper is because of the use of deferriprone on these patients. What you can see here, what is illustrated, is that the darker regions is where the ion deposition is. And what's good about this paper is that basically with beta thalassemia, it's a inherited blood disorder and you need to treat it with blood transfusions. But unfortunately, some of these patients end up having an iron overload because of that blood transfusion. But the way that it presents, what I've seen in papers, is that the black areas is similar to what we might see, especially if we're looking at the use of deferriprone. So the corresponding images below these color maps is like the intensity of how much iron is there. So particularly in what PT and CN, which is your caudate nucleus and putamen. And that is what I hope we would achieve with QSM on the brain of superficial siderosis patients. And another advantage of QSM is that it uses this de-blurring technique. So here it's not too bad. You wouldn't see like these distortions in the images, but QSM allows the influence of surrounding tissues to be removed. And that is really key to this, because in other techniques, you can't really do that. They just use a sum of everything. And then QSM, in addition to that, is way more sensitive than these other techniques. And we want that for our condition because the change takes so many years to show up. And so to pick up each subtle change, QSM has the potential to do that. So this is an experimental run of QSM, in fact, on my brain. So the top image is your standard MRI. You can see at the back where that white arrow is where that hemocydone deposition is, and that basically kind of corresponds to the QSM map that's been generated below, where you have the white arrows. So what you're looking for is basically the darker the area, the more iron deposition there is. This has all been, again, acquired via a gradient echo sequence, but it is the start of some level of work. This is the first case that we've done on my brain again, but it's the fact that we've started, and you have to start somewhere to make any kind of progress. Now, to take a further look, this is one of the algorithms that was run to produce the QSM map. This is a PDF iteratic algorithm. This was run by a postdoc fellow, but the areas circled in red, basically illustrating the level of iron, and that darkness shouldn't be there. That's not what you want on a normal brain, but that is what my brain currently looks like. There is some refining to do, because what you see next to that dark area is that blurring. Yeah, so here, this shiny whiteness that you see is that blurring, and that is a result of that magnetism from an MRI scan. So you need to refine it a bit more so you don't have that appearing. So what the postdoc quoted was that it's very noisy and it needs some refining to do. But it is still in its early stages, and there is a lot of work that has already been conducted. There has been a lot of progression in the field, particularly with a consensus paper that came out in 2024 by Professor Karen Shmuley. And she is a world-leading expert in this field and has been basically spearheading the progress of QSM. Here are just a few of her additional papers. And so with the UK government funding through Innovate UK and their KTP fund, Perspectum is collaborating with UCL, with Professor Waring and Karen Shmuley to develop this as a medical device. And it is at its early stages. And Perspectum has started a project to build on that and bring it to the patient, essentially. So with that, I bring to you Project Haemdall. I wanted to highlight this because it means that we've started, and we need to start somewhere to make any kind of progress. And we are at the cusp of beginning recruitment. It hasn't been an easy journey at all to get to this point. But since I was diagnosed, I keep saying that you have to keep pushing no matter how high the barrier is. So I bring to you Haemdall, and it may seem like a very weird name, but it is the fusion of heme, the iron compound, and Haemdall from Norse mythology, which is an all-seeing guardian watchman of the god. So in essence, I'm saying you can see all the iron. Here is our brief overview of our patient pathway. So what would happen for a patient is that you'd go and have an MRI scan. The scanning protocol has been tailored specifically for QSM and what we're trying to do. We then extract these scans from the scanner and then run them through QSM, and then use existing clinical tests to correlate the iron values, if possible, to see where that will take us. And then this allows us, hopefully, to develop that medical device. At the end of all this, what I'm really, really hoping is that all of this work will end up turning into similar products that Perspectum have that are actually FDA approved. So what would happen is that we just go for a scan. Instead of having anything invasive done, you just go in for a scan, and then that data was processed through Perspectum, and you have a report at the end, which is essentially what Perspectum does day to day, what I've been working with myself. And in particular those in the UK, where we wait forever for a report. Yeah, I see some of us laughing. We don't find out until quite some time. We could have it just downloaded from our portal, have some results, have an actual quantified amount. But this is work that will take some time, but hopefully that would be the finished product. So what does the future hold with this? Well, looking to the future direction of QSM and superficial siderosis, this lays the foundations for future studies of imaging biomarkers with clinical outcomes, which would allow for the assessment of treatment efficacy, such as iron chelators like deferoprone. You can get that quantitative data, and this is what Perspectum does with other drug trials that we're involved in. We provide that quantitative data, so why can't we do this with deferoprone or other drugs for this condition? With regards to the clinical implications, it provides objective metrics for detecting and tracking, and this allows us potentially for earlier diagnosis. And for current patients, it probably help improve disease progression monitoring and treatment response. If you have that, then you can take that data, have something in a numerical form, and use that to track everything more easily. And finally, it would help support personalized patient care plans. And with all of this, thank you everyone for your time. And if you have any questions about this, then I'm more than happy to answer. Yes, in the UK, yes. And then if it's all successful, my hope is that we can possibly take it globally. But that is, I've been told to just take it step by step for now. Yeah.
I do have a question. Do you know what is the space therapy solution? Because that is the iron is mostly in the tissue, but in superficial siderosis is mostly in the electrolytics. So I'm wondering if you might be able to capture, for example, what is happening surrounding the ender that is having the initial part of the disease.
Yeah, so on my scan, we've tried to do that, and it will take some refining because the area where the eighth nerve is, there is a lot of, what I've been told by the radiologist, a lot of blackness. So we would need to adjust the parameters and see if that is visible. But from other scans that we do in other products and seeing what has been possible, I don't see why that wouldn't be possible. Yes.
Along those lines, can the MRI distinguish where the bone starts and where the blood product is? Can you see the difference between those two signals?
So at the moment, there's a lot of artifact because of that bone tissue interface. We have been talking internally about how to overcome that issue, but there is that clear definition, and you do unfortunately have like a bit of blurring between those lines. But with some refinement of the algorithm, then I don't see why that wouldn't be possible. Professor Waring, yeah.
Basic works, it's so that you've got to this stage, and that's a wonderful talk, very, very clear.
Thank you.
Very difficult physics, isn't it? So I struggle with it. But obviously, it's going to be a really exciting thing if we can quantify the iron brain in people with siderosis. If we all agree that's a really important step. But then we've got to think about how that relates to things that are clinically important for people with siderosis. So we've got to be mindful that just measuring the iron might not necessarily be the answer to measuring the disease progression, because we've got to really understand where does the disability come from.
Yeah.
Is it to do simply with the iron, or is it the fact that some of the toxic iron is causing damage to the neurological tissue? Maybe causing volume loss, which I think we've hinted at as well. So I think this is an amazing step. and we should definitely be pursued to quantify this. I think it's going to be quite a lot of work to do in order to sort of validate it.
Yeah, there will be.
Diagnosis and as a way of monitoring the disease. We mustn't forget about other things like tissue loss, but no, congratulations on a really nice talk. My second question was just to do with have we got examples of where QSM has gone into routine clinical practice? There's certainly things to think about standardisation across devices and populations and so on. I just wondered, in your reading, have you come across any examples where it's actually made it into routine clinical?
So, what was the first question? Sorry.
The first one was just me rambling, really. Once I think if you get the quantification validated that it works and it can quantify iron, it's reproducible and all those things, and it's measuring what I think it's measuring, then I think there's a lot of work to be validated.
Yeah.
Sorry, that's really questions. I'm really interested in your thoughts though, and on the second one about implementation of this.
So the implementation of QSM, so apart from these research papers that they've tried to see if it works, I don't believe there's anything in clinic currently off the top of my head, but I'd need to double check that. And I guess with regards to validating all of this, hemosiderin is the stable compound and it's what you can measure right now. So if you determine these are just my personal thoughts, but if you determine that it's not the hemosiderin causing the issues, then you can still say that, okay, you have, this is all just theories, but if you have a low amount of hemosiderin lining the skull, but you're having a lot of symptoms, then you know that you need to go and investigate something else. So you kind of already know that there is a lot of peroxidation or free ions floating about. What I've been thinking about is how you would measure those free ions, and I know that there is a MRI phantom somewhere in the EU that actually looks at distinguishing iron 2 and iron 3. There are slightly different curves, so there is a way to check that, but obviously that would need quite a lot of work. But again, it's a start to some extent. Yeah.
How do you foresee this going into practical clinics? Do you foresee like a software that can be sold to different institutions, or like an algorithm software or something like that? Or
so I know that in the US, from what we have with liver multi-scanners, that you have CPT codes that you can order the scan itself. So initially, well, before going to that phase, you would need to obviously validate and have it all FDA approved to go to the States, which is what I think Perspectum has mastered quite well, which I don't see why there wouldn't be an issue with sorting that out, hopefully. But you would go through initially, initially validate some of this clinical data as secondary points and use something else as a primary endpoint, because then at that point the medical device doesn't need to be FDA approved if it's a secondary endpoint, if I'm not mistaken. And then you build up that data and then validate it, make it FDA approved, and then go through those channels to have it as a software as a service. And what you would do, what we currently do, is install these devices onto scanners. They each have validated parameters to set. So what we would do normally is, if we're, for instance, setting up a clinical trial, we take, and say you're dealing with a completely brand new scanner, you take the MRI phantom, which has particular parameters within their vials, run it through a scan, make sure that those parameters are fine. and then you upload the scanning protocol. And this is the case for liver multi-scan. And then our imaging applications team run a healthy volunteer to make sure that everything is running perfectly fine. And once that is fine on the scanner, you can run any patient that you need for that protocol, and have those scans uploaded and looked at. If there's any investigational findings, then a radiologist will take a look at it, or if you guys would want to download it and look at it, then that's also perfectly fine. So that would be the case of getting it as a software as a service, and medical device. So yeah, if it's possible for other organs and other devices, in my head, why can't we just do it on the brain?
Nice. Can I ask another question? Oh, sorry. Go ahead, Kyle. Two questions. First, I understand your idea about quantifying the layers of paint, but isn't it also important to know how much surface area is affected? And can quantitative susceptibility mapping detect that? Can it quantify the spread of siderosis as well as the thickness?
I would say yes, because you can get it to look at specific regions of interest and then the whole brain, which is what we're hoping to do. So
that there's an area that you'll be able to measure as well as a volume. Okay.
Yeah.
And then just really quick, have you seen any studies with quantitative susceptibility mapping using deferoprone that showed that deferoprone can remove the stable ferritin hemosiderin molecule?
Not off the top of my head. Maybe I'm misremembering, but yeah, I don't believe so. I don't think it's been done on deferoprone yet. No.
So just following on Michael's point, as I understand it, QSM will measure the total amount of iron in the brain. So it's kind of everything to do with the paint. It's how much iron's in it and where it's spread. And you get a sort of parts per million, don't you, concentration of iron in every voxel in the brain is my kind of understanding.
Yeah, so then you would, like with what we have for liver multi-scan, looking at for instance, the milligrams of iron per gram of dry tissue, you would need to have that correlated somehow. But I understand that is something that would need some development. But if there are values already available, then we can use those existing values. If there's so much research already, someone most definitely would have some milligrams of iron per gram of dry tissue. I don't see why we wouldn't be able to access that data.
No, absolutely. But I want to just pick up on your point about different types of iron. So I think this is such a fundamental question about siderosis, is whether hemosiderin is a bad guy or a good guy, because hemosiderin is the way in which the toxic iron is bound to ferritin, isn't it? And it forms hemosiderin. And you could make the case that hemosiderin is actually a molecule that's protective in some way, because it means that you are you're mopping up the iron effectively with the ferritin, and it's being deposited as this potentially very stable, could even be a harmless, stable compound. It's the free iron that's really toxic to neurological tissues. And I think this is such a fundamental point, and we might come on to it later on, Michael, in the discussions. And I'm just glad that there's a recognition that iron in itself, some of it might actually be good iron. It means that your adaptive mechanisms are neutralising the harmful free iron. But maybe Michael wants to comment on that, or other experts in the room.
I mean, my worry is that it could be in a stable form that's harmless, that then flips into a harmful state, causes damage, and then is quenched again. And this just happens over and over and over again over 20 years, and then you end up with a lot of damage. And I don't know if deferoprone can help. I mean, if you take deferoprone and it just sits in the tissue and waits for the toxic iron, is that good enough? Is it a protective agent in that way? I don't know.
I guess this comes down to the individual patient, right? If you've got a thick layer of hemosiderin and you don't know whether that patient is at the same time releasing any iron, or if you've got a patient that has a thin layer of hemosiderin and again, they might be releasing a lot of iron, but you can't see that on the brain. So I guess that if it's not hemosiderin, then this method could be used to just say, no, it's not the hemosiderin that's causing it. We can move on to something else, as like a making sure that it's not the hemosiderin, that you can use something else, basically.
Or other possibility, in our perspective, might be that the hemosiderin per se maybe is not causing the damage, but maybe what is happening is that it's upregulating through second messenger pathways the function of the microglia, the macrophages, and that maybe is causing the damage. That's other possibility that we still don't know. So
there are a lot of unknowns in this area, but it's good to start somewhere. So yeah.
Thank you. I'm kind of proud of myself because that was the question I was going to ask about the stability of hemosiderin, so it's already been asked. So I guess my point is please let us know how the SSRA can support you as we discussed earlier. Just give us a clear indication on what you think we can do today. And then number two, there's some patient day applications here. Because we have a little time, if you want to talk about your personal experience, because you did bring it up. If you don't, I understand. But if you want to talk about some of the things you've done yourself to improve your condition, because what you did put up on the screen, you look like you've done fairly well, and this has been a good presentation. So thank you.
Thank you. In terms of what I've done to keep okay, basically.
Yeah, so whatever you want to share, it's just your choice.
I'm still learning how to cope with it because I must admit, I'm a bit of a workaholic. So I know that if I work too much, then I definitely burn out much easier than I used to. I try and keep on top of my activity levels because I found, especially over COVID, if I wasn't moving, then my symptoms definitely got a lot worse. So I try and do a lot of strength training just to make sure that everything feels connected, and that I guess I also do try and strengthen each limb so that it's working on its own, basically. I rely a lot on my partner who does a lot of caring at times. So yeah, it's just figuring out what works for you. I say for me, it's making sure that I also get enough sleep. Otherwise, I'm not functioning. And if your body is screaming at you saying don't do something, don't go outside today, or you're having trouble walking, then take it easy on your body. Yeah.
Thank you very much.
Thank you.
So basically, I can just say this from my chair here. We have 10 minutes. We can take a quick break, and then we'll come back. We're going to do 2 rounds of the research roundtable, and I'm going to completely stay out of that. This will be Dr. Michael Levy's session from here on out. We're going to they'll probably do some thought and all that, and then they'll come back after a refreshment break and put some plans together. Thank you.
Oh, I mean, we have these two cameras, so we can.
Whatever case we.
Yeah, that's
Sort of towards.
yeah.
Yeah, so or the stream on.
I didn't actually.